A very big hello from me and my dearest ape friend out there. We welcome you all to evolution session where we will see how we evolved from here to here. So my dear children, with this particular video of Brahmastra series, I guarantee you can grab all the 7 marks that you can get from this chapter. There will be 2 1 marker, 1 2 marker, 1 3 marker coming from this chapter, which means we have to be very sure about giving answers for all of them. Then only we will get 7. Ma'am, after watching this video, will I get 7 marks? Absolutely, no doubt. But there's only one thing, you have to watch it fully. Do not skip even a single question. If that's okay, then let's go ahead with the first question. So the initial questions will be from the model question paper of 2025-26. And here goes the first question. Select the correct match. Australian marsupials are given on this side and placental mammals. Okay, this mammal came over here, but placental mammals are given over this side. You have spotted cuscus, all of them here. And you have to find what's the matching one. Ma'am, I do not understand what type of question is this. It's so simple. Listen to me. Okay, you have to use your imagination over here. So there was one beautiful island. Okay, this was one beautiful island. And in this island, you had some beautiful organisms whose name was Australian marsupials. Okay. Now, some of these organisms, some of them went this side and they were like, oh my God, I like that better. Some went here, some went here, some went here, some went here. All of them, if at all you see, my dear children, got elsewhere, within the same island, within the geographical area itself. And they got better adapted there. They radiated, they got better adapted there, which is why I'll say this is an example of adaptive radiation. Ma'am, don't you think it's also an example of divergent evolution? Divergent evolution, what? Divergent evolution is where you have a common ancestor, and from this common ancestor, organisms are diverging to different locations and getting adapted there. So ma'am, will they have some common features with the ancestor? Obviously, all of them will be sharing some common features. But if at all you see their functions, okay, the way they live, that's totally different. Because they have now been moved to different locations, their functions are different. Internally, they might have similar structure here and there. Okay, but they have diverged. They have diverged, and which is why this is also an example of what? This is also an example of divergent evolution. Okay, so that is about Australian marsupials. Now, one fine day, some placental mammals also came to live over here. Placental mammals, another group of organisms, also came to live over here. They also, some of them went this side, some of them went this side, here, and they went here also. Okay, so ma'am, you are saying that they also show adaptive radiation and they also show divergent evolution? Absolutely, you guys are correct. But now, that's about the individual things, Australian marsupials, placental mammals alone. But when you talk about them together in this geographical area, what type of evolution do they show? If at all you see, 2 of them moved, one from here, one from this team, moved to same location. Obviously, if at all they are staying in same location, they will have similar kind of habits also. They will have similar kind of functions also with the things, whatever they are having. They will have some commonality. Their ancestors are not same. Their ancestors are not same, but since they're living in the same location, their pretty much features somehow here and there, or basically the functions, not internal structure, but the functions, my dear children, could be same. Could be same, right? So I'll say both of them together show another type of evolution, that is convergent evolution. They have converged to a point where they have similar type of functions. They are staying in the same habitat. So when in respect to 2 of the organisms, you say convergent evolution. If you're talking about placental mammals alone, you say divergent. If you're talking about Australian marsupials alone, you say divergent. Ma'am, why are you talking about all of this? The question does not ask this, right? Array, it asks. So you are being asked, from Australian marsupial side, you have spotted cuscus. You have what? Spotted cuscus. What is there as of match the following in placental mammal side? That is the question. So your answer for this is spotted cuscus and lemur. Ma'am, how will we remember this? It's super simple. So listen, this is the chart given in your NCERT. Placental mammals' names are very short. On the other hand, Australian marsupials, my dear children, their names are a little big. So over here you have mole, there you have marsupial mole, easy. Anteater, numbat. Numbat is also the other name of anteater from Australian marsupial side. Okay, that's a different name that we have. Mouse, marsupial mouse, easy. Okay. Lemur, spotted cuscus. This is exactly what we studied right now. Flying squirrel, flying phalange. Flying squirrel this side, flying phalange that side. Bobcat, Tasmanian tiger cat. Wolf, Tasmanian wolf. The names are bigger over here. Flying squirrel and flying phalange also might be difficult for you to remember. But remember, over here, phalange starts with P, but it does not come under placental mammals. It also starts with P, but it does not come under placental mammals. It comes on this side, opposite. You remember like that. Now, the names that are different and you need to remember, be extra cautious, is anteater and numbat. So, from placental mammal side, you have anteater. From Australian marsupials, you have numbat. You have what? Numbat. Lemur from placental mammals, spotted cuscus from Australian marsupials. Right? Okay. Flying squirrel from here, flying phalange from there. So, this side, so all these three, 1, 2 and 3 have different names. Rest all of them have, what do you call, kind of similar names so that you can remember it easily. Okay? Done? I hope this question is super clear and you will be able to attempt any questions that come from here. Yes? Yes. If at all they're asking you, mole, anteater, mouse, lemur, squirrel, bobcat, wolf, all of them evolved from the common ancestor. What type of evolution does it show? It is divergent evolution. All of them had what? A common ancestor. Now they're saying mole, marsupial mole came, anteater and numbat also came. What type of evolution does it show? It is convergent, because here you're talking about two different groups of organisms. Okay. Children, understanding? Liking the video? Yes? Then what are you thinking? Click on the like button. Share it with your friends. Okay. Now, which of these ancient men protected their body with hide and buried their dead? Which of these ancient men? For that, we should know the whole story of how men were evolved. Okay, how organisms, man, how were they evolved? So first of all, I'll tell you the story. Why is this question important? Because it can come in a 2 marker, 3 marker, any format. So make sure when I talk about this, you listen to it as a 3 marker question. The maximum amount of marks should be, you should be getting it from here itself. So initially, my dear children, we had 2 types of organisms. One is Dryopithecus, okay, and there is Ramapithecus. I'm not checking the spellings over here, okay. Rama has a term Ram, so you can remember it was more human-like. This was more human-like and this was more ape-like, okay. Dryopithecus were more ape-like, that was more human-like. So let's talk about them then. If they were human-like, obviously they are going to evolve, yeah. From here we got Australopithecus. And the first thing that was noticed about them is that basically they don't eat meat, rather they were basically eating fruits and stuff like that. Okay, fruits and stuff like that. From Australopithecus, we got the first human-like hominid. For the first time, I saw a first human-like hominid out there, which is why I'll say it is Homo habilis. Yalla habibis, we say no habibi. So Homo habilis we got from there, and from Homo habilis we got Homo erectus. Now they started standing erect like this, okay. So Homo erectus. From Homo erectus, my dear children, you got Neanderthal man. You got 2 things. First thing that you got was Neanderthal man. So until here, until Homo habilis, all of them were eating fruits and all. From Homo erectus, they started eating meat, and then came Neanderthal man, and just after that, you got Homo sapiens, that is the modern day human beings. So first came them, then came us. Now over here, as I said, there are some very important factors that you need to remember. First, human-like hominid that you got was what? Habibis, Homo habilis. Homo erectus were the one who started eating what? The meat and everything. They switched from fruits to meat, okay. And Neanderthal man were the first ones to basically bury their dead. That means they started understanding relationship, they started burying their dead, and they also started hiding, covering their body as well. So they covered their body and they hid their dead. That was very, very important. The Homo sapiens, so you know, that is us. So which of the ancient men protected their body with hide and buried their dead also? It was what? Neanderthal man. It was what? Neanderthal man. Ma'am, we studied this, that's enough, right? No. So there is a whole paragraph that I have written over here, not me. So basically, I have not written, it's from your NCERT itself. You need to remember each and every detail of it, because as I said, a 2 marker, a 3 marker can show up anytime. So few very important things that you need to remember for sure is years. So 15 million years ago, how many? 15 million years ago, you got Dryopithecus and Ramapithecus. Ramapithecus more man-like, Dryopithecus more ape-like. That means we are going to focus on this guy. Now 3 to 4 million years ago, after 15 million years ago, 3 to 4 million years ago, you got man-like bones, fossil evidence from Ethiopia and Tanzania. Okay, Ethiopia and Tanzania, my dear children. Height was almost around 4 feet, adapted to bipedal locomotion. That means there were 2 legs and they started moving with that. Usually we were walking like this, no? Initially. But now, bipedal locomotion was seen 3 to 4 million years ago. So that's when the fossils and everything was recovered. Now, from Ramapithecus directly, we come to Australopithecus. That happened 2 million years ago. That happened 2 million years ago. So 15, then 2. Remember like that, you will be asked match the following question. This and this match. When did they come? Australopithecus, it was 2 million years ago, lived in East African grasslands, used stone weapons, okay, main diet was fruits, not primarily meat eaters. They were not meat eaters. Now, after that, you had Homo habilis. For Homo habilis, you do not have ear being mentioned. So, we assume 2 million years ago, you have Australopithecus. From them, normally, you got Homo habilis, the first true human-like hominid. This is a very important feature. And from this point onwards, you have your cranial capacity is being mentioned in the NCERT. That means your brain capacity. Cranial, CC stands for cranial capacity. So, the cranial capacity is 650 to 800. Over here, this is the first one. Now, you will see gradual increase in the cranial capacity. We are developing, right? So 650 to 800 cc was there in Homo habilis. There was the first human-like organisms out there. They used simple tools, did not consume meat. So from stones they came to tools, but here also they did not consume meat. Now after 2 million years ago, you have 1.5 million years ago Homo erectus. The brain size increased to what? 900 cc. So here it was 650 to 800. Now you have 900 cc. They started using fire, crafted better tools. Diet included what? Meat. And fossils were found in Java, Indonesia. Their fossils were also found in what? In Indonesia, in Java, my dear children. They found the fossils of Homo erectus. That means they evolved there. Yeah? And now after 1.5 million ago, almost 1 lakh to 40 years ago, 40,000 years ago, my dear children, you got Neanderthals. Brain size was the highest. It was 1400 cc. So from 650 to 800 to 900, now to 1400 cc, larger than the modern day human beings, larger than us actually. Okay, they lived in caves, used animal hides for protection, buried their dead as well. Found in Europe and Central Asia. So from Indonesia to Central Asia, they moved around here and there, my dear children. And almost 75,000 to 10,000 years ago, that is the recent most time, Homo sapiens, you got the modern humans. They were originated in Africa. You need to understand that. Then migrated worldwide. Developed language, culture, social structures, everything. So please remember the years when you got them. From Homo habilis, you have the cranial capacity being started off. Please remember that as well. Rest everything. I have told what's important, right? You just need to remember that. So if at all you're asked, the fossils of which organisms were found in Java, you should know it is what? Homo erectus. It is Homo erectus. Okay, Europe, Central Asia, it's them. But usually questions have not come like that. Usually it's the ears and the features. So please remember them. And the most common thing that is asked is Neanderthal man. 1 lakh to 40,000 years ago. Please remember that also. Okay, and here 75,000 to 10,000 years ago. Cool. Now, name. This is a 2-marker question. That is, okay. Name the evolution of any 2 plant forms through geological time periods from tracheophyte ancestors. From tracheophyte ancestors. Ma'am, I do not know. This is very difficult for me. Easy. So, over here, I'll tell you a story, okay? So, initially, the plants which were formed, we say they were algae. They were categorized as what? Algae. They did not have root, stem, leaf, nothing. Okay. From algae, you got the bryophytes. You know, bryophytes are the ones which will be having root-like, leaf-like and stem-like structures. They did not have it properly, but they had root-like, stem-like, leaf-like structures. And before I go ahead, algae were the ones. Why did you put it under plants if they do not have root, stem, leaf, anything? Because they had chlorophyll pigments. They had what? Chlorophyll pigments. Okay, that was very important for photosynthesis to happen. Now from them came bryophytes who had root, stem, and everything like features. They did not have proper thing. They had root, stem, and everything like features out there. From Bryopithecus, you got. Sorry, not Bryopithecus, oh my god. Bryophytes, you got pteridophytes. You got pteridophytes. Now, pteridophytes are the ones who, for the first time, had roots, leaves, stem and everything. Apart from that, my dear children, they had vascular system, as in there was xylem and phloem, and xylem for transport of water and phloem for transport of food. So, the vascular system came up over here in pteridophytes. Now, from pteridophytes, you got gymnosperms. Gymnosperms are the one which produces seeds, but they do not have flowers. They produces seeds, but do not have flowers. From gymnosperms, what did you get? You got angiosperms, the flowering plants. The flowering plants. Here, the seeds were covered by some fruit and things like that. So, this is the flow. Why did I tell you this flow? Because I want to explain you this particular thing. You have been asked to tell the evolution of any two plant forms through the geological time periods from tracheophyte ancestors. When you're asked about tracheophyte, understand you're talking about pteridophytes, because from here you got the vascular system. Trachea, which is there, it's a part of the xylem. It's a part of the xylem. So here is the thing that is being asked about. So from tracheophyte ancestors, how do we say? So let me come over here. So chlorophyte ancestors, as in I told you, it's the algae and stuff like that. From them you got tracheophytes. Ma'am, bryophytes, where are they? So over here you see one line going up. One line, if at all you see going up, it is bryophytes. Okay, these are the bryophytes, but they did not come from tracheophytes. You have been asked about what's come from the tracheophytes. So let's see. From the tracheophytes over here, you got zoosterophyllum, arborescent lycopods, herbaceous lycopods and things like that. If at all we go ahead, we get Rhynia type plants. From them we get Xylophyton. So have this in common. Okay, have this in common. Tracheophyte, then Rhynia type plant, then Xylophyton. From Xylophyton, you get the modern day ferns, my dear children. The spinopsids and the ferns are basically the actual pteridophytes that you're talking about. The actual pteridophytes that you're talking about. Okay, now if you feel that, ma'am, I should not be talking about pteridophytes because anyway I started with pteridophytes, right? Let's talk about gymnosperms and angiosperms. Can we tell that? Yes. How can we tell that? So from here, we got this. From here we got Xylophyton. From Xylophyton, if at all you see ginkgos, conifers, okay, ginkgos, conifers, all, what are they, my dear children? They are gymnosperms. They are the gymnosperms. So this is one thing that you can write: ginkgos, conifers, knee-tails, cycads, all of them, or basically just write this: ginkgos and conifers and knee-tails, all of them are basically what? Gymnosperms that you got. Now we need one more thing: angiosperms. How did you get it? From here, tracheophytes, Rhynia type, Xylophyton, again progymnosperms, then seed ferns. From seed ferns you got dicots and monocots. They are basically angiosperms. Dicot first and monocot second. They are the angiosperms. So this is what you have to write. How do you write it? Okay, tracheophyton ancestors, then Rhynia type plants, then Xylophyton, then you got ferns. That is basically when you're talking about pteridophytes. If at all you want to write about gymnosperms, the same thing, but instead of ferns, write ginkgos. Because ginkgos, which is there, it is a gymnosperm out there. Okay, you can write like that. Now angiosperms, tracheophyte ancestors, Rhynia type plants, Xylophyton. Now progymnosperms, then seed ferns, then dicots. Okay, the dicots and everything out there. Or basically you can directly write the angiosperms out there. So this is how you need to write. And for that, you need to know about them. Always remember the gymnosperms, ginkgos, conifers, knee-tails and cycads, they were over here in the Mesozoic era towards the Jurassic and Cretaceous time period. It's very, very important. Similarly, if you talk about the pteridophytes, this and this pteridophyte is super famous. Spinopsid and ferns. They are also Cretaceous, Jurassic. Jurassic, Cretaceous era. Similarly, the gymnosperms also in the same era. Angiosperms, dicots came over here. Then came your monocots towards the Cenozoic era. The next era. So this is Mesozoic, this is Cenozoic era. The lower one is Paleozoic. Paleozoic, Mesozoic, Cenozoic. Okay, here you have, this era is very, very important because here you have most of the organisms being formed. Towards the end, you also have angiosperms being formed. Cool? Do not forget it. Jurassic, Cretaceous. Jurassic, Cretaceous. Of which era? Mesozoic era. Okay. Now draw a labeled diagram of Stanley Miller's experiment in relation to chemical origin of life. Now this question is repeated in question MQP1, that is model question paper 1, and also model question paper 3, question number 31. Okay, so question number 31 of these papers are the same only. Stanley Miller's experiment. So what was Stanley Miller's experiment? He was trying to prove what Oparin and Haldane said. Oparin and Haldane said that basically there is chemical evolution that has taken place. All the organic matter that you see has come from the inorganic matter, and this organic matter which are formed from the inorganic matters, they also come together to form big molecules and things like that. That's how the earth evolved, is what they said. Okay, so if at all you were not able to recollect, I'll write it. So they said there was inorganic things. These inorganic things came together to form some organic molecules. These organic molecules, my dear children, combined in different format to give what? A proper cell and structures like that. Okay, that is what is said in chemical evolution. Stanley Miller came here to prove that. That yes, from here to here, from inorganic, you can make organic stuff. In accordance with the primitive earth. He made a setup like this, and with that he tried to prove. So over here in the setup, if at all you see, my dear children, you have boiling water. Why do you need boiling water? Because we need water vapor. Water vapor was there in the primitive earth, so we need that. So water vapor is going to go up over here. If at all you see, it's closed chamber. Why is it closed chamber? Because in the primitive earth there was no oxygen. Remember, no oxygen was there in the primitive earth. It was a reducing environment. When there is no oxygen, you say it's a reducing environment. Okay, so no oxygen, water vapor is there. Why do you think electrodes are kept over here? Because the temperature was super high. Earth was molten mass. 800 degrees Celsius is being maintained over here. So for maintaining that, you need electrodes. Okay, for maintaining the same temperature, you need electrodes. Now, there were some gases in the primitive earth. NH3, that is water here. So I'll start with methane. CH4, that is methane. NH3, ammonia. H2O, water vapor, and H2 as a gas. Okay, methane, ammonia. Methane is CH4, not CH3. Okay, methane, ammonia, H2O as in water vapor that we get from here, and H2. These gases were there in the presence of all this thing, that is reducing environment, 800 degrees Celsius. What is happening? If at all you see, some organic molecules were accumulated over here. They were formed over here, they're getting accumulated over here. What was this organic molecule that Stanley Miller found in his experiment? It was nothing else but just amino acids. What? It was nothing else, just amino acids. Amino acids are the building block of proteins. When amino acids comes together, my dear children, that's when you get protein. Always remember, there are several other things, for example, nucleotides, this and that. You did not find anything in Stanley Miller's experiment except amino acids. Okay, so this is the thing. Remember, 800 degrees Celsius, super important. Reducing environment, as in there was no oxygen. And the name of the gases. Okay, it was CH4, NH3, H2 as in water vapor format, and H2. Done? Done. Next. Arrange the following animal groups in correct sequence of their occurrence on geological time scale. This is model question paper 2, question number 6. So you're given the options of organisms, amphibians, fishes, reptiles, and mammals. You are being asked, please arrange them in order. So, all of them are vertebrates. That means they have vertebral column. This thing, vertebral column is there, my dear children. So first of all, we had fishes which were there in the water. Now some of them evolved and said that I do not want to be in the water, I want to be on the land. So from them came amphibians. Amphibians as in they can stay in water as well as land. Now from amphibians you got the reptiles as in the snakes and things like that. From reptiles you got two things, mammals and aves. Mammals and birds. Aves as in birds. This is how the thing goes. So fishes, amphibians, reptiles, mammals and aves. So if at all you want, F-A-R-M, farm. Okay, farmland, you can remember like that. Farmland, fishes, amphibians, reptiles, mammals, and over here, my dear children, aves is also there. Please remember, both of them evolved from reptiles. So in that case, 2 should be coming first. 2 comes first. I have 2 options like that. Let me cut down the other options. Okay, after fishes you had amphibians, 2 after that you have 1. So this is your answer. 2, 1, 3, 4. 2, 1, 3, 4. Got it? Yes. Next, The paleontological evidence for evolution is obtained from paleontological evidence as in basically evolutionary evidence in the form of fossils. Okay, from the fossils, how, for example, when I go deep into the soil, I find the fossils of this organism out there. When I go here, I find another organism out there. So basically, these fossils gives me an evidence which is known as paleontological evidence. At which era, what organisms were living, we get to have an understanding about that. So the paleontological evidence for evolution is obtained from fossils. This should be your answer for the question. Okay? Okay. Next question. Comment on the similarity between the thorn of bougainvillea and tendril of cucurbita with reference to evolution. Okay, we have two plants. One is bougainvillea. Okay, this is bougainvillea and the other one is cucurbita. So over here you have a thorn and over here you have a tendril. What if I tell you that basically both these structures which is there have evolved from the same, okay, same basic structure. They have both evolved from the same basic structure or the anatomy, that is axillary bud. They evolved from the same structure. They are now at two different habitats. One needs support, the other needs protection, which is why both have developed differently. So what do I say? This is an example of what? Divergent evolution. They diverged, my dear children. This is an example of divergent evolution. And what do you call these type of organs which grow different, but internally they have evolved from the same part? What do you call them? This is homologous structures. Homologous structures. Listen, the examples for homologous structures is very, very important. For convergent evolution also super important. Please remember this. So I remember like this: homologous organs show divergent evolution. That means HD. H for homologous, D for divergent. Similarly, convergent evolution means analogous organ. Convergent evolution we studied, no? Remember, two organisms from different groups coming together. I'll give you another example. You have this thing, birds of butterfly and birds of bird. Wings of butterfly and wings of birds, my dear children. Both are wings, both are used to fly, right? Both are used to fly. But if at all you see the structure, is it same? No. But they are living in the same habitat because why? They are converged to be like that. That is convergent evolution. They do not have any common ancestor. But over here you had shared common ancestors, which is why you had similar structures. From these similar structures you have evolved something else which have different functions. That is divergent evolution. There it is convergent evolution, and the organs which are there, the wings of the butterfly and the wings of the bird, they are what? Analogous organs. So A, C, H, D, you can remember like that. Okay, similarity between the thorn of bougainvillea and tendril of cucurbita. Let's see, how do you write the answer? Both the thorn of bougainvillea and the tendril of cucurbita are homologous organs. Both originate from the same basic structure, that is the axillary bud or the meristem. Okay, but perform different functions. Thorn is for protection, tendril is for climbing or support. Evolutionary significance. The similarity indicates divergent evolution where structures with common origin evolved to perform different functions due to what? Adaptations to different habitats. Okay, and with this, since we're anyway discussing this, let me just move to that question. I had that question here itself. Just a second. Okay, what are analogous organ? Give an example. So as I told you, analogous organs are the one that basically have similar function but have different origins. They do not have any common ancestor. They show convergent evolution, such as wings of birds and bats and insects. All of their wings are different. For birds, it has bones and everything. You cannot break it off. Bat again, you have different structure out there. Bird is under aves, bat is under mammals, and insect is a whole different category itself. Okay, so but all of them have wings to fly only. The function is same, the structure is different. Convergent evolution, since they are staying in the same habitat. Eye of octopus and that of the mammal. Eye is there for seeing things. But if at all you see the structure of the eye of the octopus, it is very different from that of the mammal. Both of them functions are same. Always, if at all you want to find convergent evolution and analogous organs, see if the function is same. Eye of octopus and mammals is the same, only function is to see, but the structure is different. Flippers of penguins and dolphins. Flippers of penguins and dolphins, my dear children, flippers out there. In the case of penguins, What is it for? In the case of dolphin, what is it for? Both of them are using it for the same function, that is swimming. That is swimming. But dolphin is a mammal, penguin is a bird. Dolphin is a mammal, penguin is a bird. Okay, do not say dolphin is a fish. Dolphin is a mammal and penguin is a bird. Now, root modification of sweet potato and stem modification of potato, my dear children. Potato, sweet potato, both of them are using their structure for storage. Function is same. They are using the structure for storage, but do you know that both of them are evolved differently? So over here, if at all you see sweet potato, you might have seen the plant. Plants have roots, right? Suppose the roots are modified. This modified structure that you see is known as the sweet potato. The modified root that we have is known as sweet potato. On the other hand, if the stem, my dear children, it grows underground. It grows underground, you have another root separately. The stem grows, it is known as potato. The stem grows into a structural format that is known as potato. So that is a stem modification. Potatoes is a stem modification. Here root is getting modified to form the sweet potato. Function is the same, that is to store. So they come under examples of analogous organs. They come under examples of analogous organs. What type of evolution do they show? Convergent evolution, AC, remember that. Okay, now we will go back to the same question. Where are we? We are somewhere over here. Yes. Okay. Now, observe the following diagrammatic representation of operations of natural selection on different traits here. Natural selection depicts. So here different type of, there are different type of natural selection. One is this, the other one, there are two more. So suppose you have a feature that is height is tall and height is dwarf. In between that you have a feature that is medium heighted organisms. Suppose all the organisms are tall. That means it is directional, either on one side, either tall or they are dwarf. Either tall or they are dwarf. This is known as directional selection. Any one extreme is being selected. Only one particular extreme, my dear children, that is being selected. Where does it go? Okay. Now, suppose it's something like this. Same thing. You have tall, you have dwarf. Both of them are selected. Some people are tall, some people are dwarf. This is known as disruptive selection. Two extremes are being selected. This is known as disruptive selection. Now you have one more. What is that one more? So here you have tall, here you have dwarf. Okay, here you have tall, there you have dwarf. Now, out of them, no one is selected, but the median character is selected, that is a medium heighted. This is known as stable selection, stabilizing selection. This is known as stabilizing selection. So either it can be this side or this side, then it is directional, only one extreme is being chosen. If both the extremes are chosen, then disruptive, and if in middle, then it is stabilizing. Okay, here you are given two of them, two curves are there. That means it has to be disruptive. This is going to be disruptive selection. Okay, okay, ma'am, done. Now, dash stage in human evolution used height to protect their body and buried their dead. Again, the same type of question is being repeated. Which organisms protected their body? Neanderthal man. What was the cranial capacity? 1400 cc. When did they evolve? 1,02,000, 40,000 years ago. So all these things should be coming in your head. Okay. Next, state the evolutionary relationship giving reasons between sweet potato and potato. We just discussed it. This is model question paper 3, question number 25. So, sweet potato is modified root, whereas potato is modified stem. Both store food but differ in origin and structure. And if origin is different, they show what? Convergent evolution. Such organs with same functions but different origin are called analogous organs, and their occurrence is due to convergent evolution, where unrelated structures evolve similarity to perform the same function since they are staying in the same habitat. Since they are in the same habitat. Okay. Cool? Works? Yes, ma'am. Fine. Now, question number 11. This is question number 31 of model question paper 3. With this, I think we are ending all the model question paper questions, right? Australian marsupials and Australian placental mammals explain Convergent evolution and adaptive radiation. Justify the statement. Okay, so Australian marsupials and Australian placental mammals, they show convergent evolution and adaptive radiation. Justify the statement. I already told you, if two organisms of different ancestors, they are evolving, okay, from them, from both of these category, one organism is coming to a location, my dear children, they are showing convergent evolution. So how do you write it? You write like this. In Australia, many marsupials evolved from a common ancestral stock and diversified into different forms, adapted to different habitats and functions in a geographical area. This is an example of what? Adaptive radiation. Now over here, placental mammals in Australia evolved independently but developed structures and habitats similar to that of what? The marsupials, to occupy the similar ecological niches, to be in the same area. This similarity is due to convergent evolution. So first, with the help of marsupials, you can explain adaptive radiation, that basically from a common ancestor, they diversified into different forms within the same geographical area and they got adapted there. That is adaptive radiation. Now, within the same place, placental mammals independently also did the same thing. Okay, they also basically developed different structures and habits, but wait, how do you frame this? But developed structures. Okay, yeah. So basically, Australian placental mammals also came into the picture, and what did they do? Independently, they developed same habits, just like same structures and habits, just like the fellow marsupials out there, because both of them are staying in what? The similar habitat or the same habitat. You also say same ecological niche. This similarity is due to what? Convergent evolution, or this similarity shows what? Convergent evolution. Cool? Yes. Give examples of few of them and then it will be a lot more better. Cool? Cool. Now the revised question bank questions, most of them are going to be MCQs, my dear children, because with MCQs we can understand a lot of concepts as well, right? So statement 1 says, man has domesticated dogs and has created different breeds by artificial breeding. Is that true? Has man domesticated dog? Yeah, we pet dogs, right? And actually with the help of artificial breeding, we have created so many breeds out there. This is there in your NCERT, this is correct. Intensive breeding of dogs signifies divergent evolution. Intensive breeding of dogs signifies divergent evolution. They were having a common ancestor, we diversified them, my dear children. So yes, they had a common ancestor, they moved to different locations, had different habitats. If you see a husky, it looks different. If you see a normal dog, Indian dog, it looks different. Why? Because they are in the they're in a different habitat. But all of them have some common features because that's an example of divergent evolution. So can I say both statement 1 and 2 are correct? Yes, ma'am. I can say that. Okay. Next. Statement 1, antibiotic or drug resistance is due to evolution by anthropogenic actions. What? So basically, suppose in our body, okay, in Pappu is my friend, okay, in Pappu's body, Pappu had an infection with some bacteria out there. He usually used to have so many antibiotics. Now what will happen? The bacteria which is there, it will say that, these antibiotics are not going to work on me because I have resistance to it. Since you have so much of it, I have now found ways in which I am resistant to certain antibiotics. Why did this happen? Because of our nature, my dear children, because of our action. And these actions are known as anthropogenic actions, the human actions which leads to problems. So, antibiotic or drug resistance is due to evolution by anthropogenic actions, yes. Many antibiotic or drug-resistant organisms arise due to over-usage of antibiotics. Yes, Baba, it does. And that's exactly what we are doing. Right, so this is an example of evolution by anthropogenic action. Please remember it. Anthropogenic as in man-made actions. So both statement 1 and 2 are correct. A is your answer for this particular question. Okay? Done? Deal? Chalo. A brown-billed duck is newly added to a farm of white-billed ducks. This indicates what? So over here, I had the white-billed ducks out there. Okay, the white ones were there. Now, what I am doing? I am putting some brown-billed ones also. What does this indicate? Gene flow, genetic drift, mutation, natural selection. It's gene flow. Because from elsewhere, I had taken this organism and put it over here. When I put an organism over here, obviously that organism has some genes out there, some alleles out there which gives it the brown color and everything. Am I not transporting the genes also? Now when these organisms, they mate, basically there will be exchange out there, there will be newer combinations being formed. Yes, so this is what? Gene flow that is happening. Gene flow that is happening. Remember, this is a factor that affects Hardy-Weinberg principle. Hardy-Weinberg principle says that the allelic frequency or the genetic frequency is going to remain the same. Is going to remain the same. For example, I have tall plants. Okay, tall is represented by capital T. No, I have so many tall organisms out there, and I have so many dwarf organisms out there. Few of the dwarf organisms and so many tall. Now, according to Hardy-Weinberg principle, he says that the allelic frequency, the frequency of capital T allele and the frequency of small t allele is going to be the same. It is going to be in equilibrium over generations and generation. But do you think that can happen? No. Suppose some short people came into the picture. So that means the allele frequency of small t is going to increase, right? So there are factors that affects the allele frequency. One such factor is gene flow, where from here genes are going to get migrated, as in when people, they move around, organisms when they move around, it causes the gene flow. Okay, genetic drift is another thing. Genetic drift means, suppose over here, I'll explain everything. So genetic drift means over here, you have some organisms. Okay, you have some organisms out there and they were like, we are the ones who are dominant, you are the ones which are recessive. One day, one big elephant came and stamped on them and all of them, they died. All of them, they died. What will happen? Now, who is left behind? The white ones. So, just by a chance, a random chance, some alleles, my dear children, their frequency got away, and now the frequency is totally different. This is genetic drift. A drift is happening. Mutations, so you know. So, suppose over here you just had the white ones and you had the brown ones. Some mutations happened in their genes, my dear children, which is why now you have the green ones. Allelic frequency, don't you think it is going to change? Yes, it is. Natural selection. Natural selection example, my dear children, you all have studied about it. Okay, there was white moth, black moth, things like that. If at all you do not recollect, we will talk about that once more also. Natural selection is when nature is selecting. Nature is going to select. I'll explain it now only. Yes, I'll explain it now. Listen to me carefully, okay? So over here, I have two era. Before industrialization, that is before industries were formed, after industries were formed. So before industries were formed, the whole plant, my dear children, it was covered by lichens. Lichen is a combination of fungi and an algae. Fungi and algae. And they can only grow in areas which are very clean, neat and tidy. Okay? After pollution, obviously, nothing is neat and tidy, which is why lichen cannot grow. So I can say that lichen is what? A pollution indicator. This is also a question. Pollution indicator. Okay, lichen is not going to grow there. Now, you had two type of moth. One is the white one and one is the black one. Since black color is not visible, I'll make it as blue. Okay, black and white moth were there. Black one arose because of some mutation, which is why that's how you got the black ones. Now, my dear children, which one do you think will be able to survive over here before industrialization? The white ones. Because the white ones, if at all they sit on the bark and here and all, no predator is going to see them. But on this background, if at all you see a blue one sitting, my dear children, what is going to happen? Obviously, they can be easily trapped. So not blue, but the black one. You get it? So which one is going to survive better? It is this. So nature selected the white ones before industrialization. So the white ones increased the number, whereas the black ones, their number was very less. After industrialization, now suppose over here, if I put a black organism, will you be able to see it? No. So what about the white one? If I put a white organism, the predator will be easily able to catch it, right? So now, what is the nature selecting? Nature is selecting this one. So allelic frequency is going to change according to what? The nature. This is known as natural selection. The nature selects it. And this is also an example of evolution by anthropogenic action, because it was man, my dear children, who made sure that there is pollution and because of which all of this happened. But anyway, so a brown-billed duck is newly added to a farm of white-billed duck. This indicates what? Gene flow. There was a group of organism, you are inserting something, that means a gene flow is going to happen. Easy. There is no genetic drift happening up over here. Okay, there is no mutation, there is no natural selection. This is gene flow. Next, change in frequency of genes and alleles is due to. Change in frequency of genes and alleles is due to mutation, yes. Gene flow, yes. Both A and B, yes. Neither A and B, wrong. So you should know all the factors. Now there are multiple factors, we will be talking about that. I have a bigger question, I will be showing you that question as well. And remember over here, lichen is a pollution indicator. It cannot grow where pollution is there. Next, the first amphibians that lived both on land and water evolved from. The first amphibian that lived both on land and water evolved from. What is the answer for this question? First amphibians that lived both on land and water evolved from fishes, turtles, dolphins, frogs. Fishes, turtles, dolphins, frogs. Ma'am, you said that fishes give rise to amphibians. No, so this is the answer. Baba, please understand. In fishes, you had a type, okay, dolphins which had big fins and they could move with their fins on the land. So this is like a transition that we have from fishes to amphibians. From fishes to amphibians. So your answer for this question is lob fins. The first amphibians that lived both on land and water evolved from the lob fins. If you have a specific option like this, please remember, it's a very important link between fishes and amphibians. You get questions like that. Lob fins, do not forget them. Okay? Now, the biggest land reptile that existed during the Mesozoic era was the biggest land reptile that existed during the Mesozoic era. What? Mesozoic era as in you have Jurassic era and things like that. Jurassic season, you have watched the Jurassic Park movie, in that you have so many dinosaurs, right? So over here, the biggest land reptile that you have is Tyrannosaurus rex. Tyrannosaurus rex, my dear children. This is your answer for the question. Tyrannosaurus rex. Okay? Statement 1. Hugo de Vries believed mutation leads to spontaneous evolution. So, you know, Darwin was doing his experiment. He was saying that evolution is a gradual process. Listen to me carefully. Darwin said evolution is what? A very gradual process out there. Okay, and it happens due to variations and things like that. Hugo de Vries came into the picture and said, you know what? Mutation is the cause of evolution, and mutation, my dear children, is a spontaneous thing. Okay, mutation leads to spontaneous evolution, not gradual but spontaneous evolution, and mutations are going to be random. Okay, it can happen anytime, anywhere, it can be anything, and they are directionless. It does not say that okay, this mutation is going to be good, it can be bad, anything. They are directionless and causes saltation. What is saltation? This is a very important thing. Single step large mutation. Single step large mutation is what? Saltation. And with that you got evolution. You get evolution, a new organism evolving out there. So Hugo de Vries believed mutation leads to spontaneous evolution. Yes. He was against gradual evolution, which was told by Darwin. Okay, Darwin didn't have much idea about mutation and all. He said some variation was there, and organisms which had better variations, they were able to adapt better. That is, the fittest one was able to survive. They were able to reproduce also. And what was his name? I forgot the name of that person. As of now, I'm not able to remember. Anyways, so yes, and mutations are random, directionless, and causes saltation. Single step large mutation is known as saltation. All of it was told by Hugo de Vries, and all of it was correct. So I'll say both 1 and 2 are correct. Remember, both of them had different opinions. Both of their opinions were actually correct also. Okay, elongation of neck in giraffes due to inheritance of acquired traits was given by. Elongation of neck in giraffe due to inheritance of acquired traits was given by. So there was a guy whose name was Lamarck. Lamarck said, you know, there was the giraffe had shorter necks, but their food was at the top, which is why they did like this. And when they did like this, my dear children, that it was naturally inhabited in their genes, and it was transferred to the next organisms they were reproducing. But do you think acquired character, that means something that we acquire during our lifetime, that can be passed to next generation? No, we cannot. For example, there is an experiment that was proved to done to prove that whatever he's saying, it's wrong. So a mouse was there, okay, it had tail. We cut the tail of this particular mouse. Now what do we expect if this is an acquired trait now? What is an acquired trait? The mouse without the tail. We expect this trait to be passed on to the next generation. The next generation should also not have tail, but over here they had tail. That means acquired traits are not going to be passed on. But this was not something that Lamarck said. Lamarck said the opposite of it. He said that the acquired traits are passed, and that's why there is elongation of neck in giraffe, because one organism did that, which is why it is being passed like that. So that's what Lamarck said. Whereas Charles Darwin, Thomas Malthus was a person who came along with Charles Darwin and basically said that Thomas Malthus' works inspired Charles Darwin, okay? Thomas Malthus' works inspired Charles Darwin, and he said that, yes, you're correct, the one who has variations, they are going to adapt better, they're going to reproduce and things like that and everything. It's a very gradual process, blah, 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 blah. Hardy Weinberg, on the other hand, told about mutation, I told you, right? All of their things were correct, but Lamarck was wrong. Lamarck was absolutely wrong in this condition. Okay. Next, single-step large mutation leading to speciation is called. Single-step large mutation leading to a new species formation is called saltation. Is known as saltation. I told you, it's important. Please remember it. Yes. Now, the organic compounds formed in Miller's experiment are. What are the organic compounds formed in Miller's experiment? Amino acid, nitrogenous bases, pigments, fats. Only one thing, and that is amino acids. I already told you when I explained about Miller's experiment, there is nothing that was found in Miller's experiment. Other people did similar experiments. That's when they found the nitrogenous bases and everything. As of now, it's just amino acids. Okay. Embryological evidence for evolution was disapproved by. Okay, so we know paleontological evidence, that is done by fossils and all. You have radioactive dating and all these things. That is paleontological evidence. Embryological evidence basically means, suppose, my dear children, there was a guy whose name was Ernst Haeckel. He came up with this evidence. He said that, we studied that fishes are there, then we have amphibians farm, right? And then we have reptiles. And then we have mammals and also aves. So basically, in mammals, when we are in embryonic format, we also have fins, we also have tail. Yes, we do have that. But when we evolve in the womb, my dear children, all of it goes away. But we have those features. So he said, probably the organisms from which we have evolved, okay, their features will be shown in the embryo. That means we might have evolved from those organisms. That was told by Ernst Haeckel, which is why we have fins, we have tail and everything. That is embryological evidence. To say that it's no, a person with a similar name came into the picture, Carl Ernst von Baer. Carl Ernst von Baer, bigger name, okay, he said no. He said in that case, it should show all the features. It should show the features of amphibians also, should feature some reptiles features also. If we have evolved from them, you should be seeing their features also in the mammals, right? Why it's not like that? Why only certain features of fishes are being followed, that is gills as well as tail and things like that? Why not all the organisms, some features are being copied up there? That's not happening. So I'll reject it. So it was rejected by Carl Ernst von Baer, but it was put down by Ernst Haeckel. And both of them were partially correct in their own ways. Okay? Now, arrange the following plants as appeared on different geological periods. Now, how will you be able to answer this if at all you know this particular thing? How do you arrange the following plants in geological time periods order, my dear children? You have conifers, you have knit-tails, then dicots, then monocots. The first option itself is correct. Conifers, knit-tails, dicots, monocots. So remember CC together, GG together. Ginkgos, knit-tails over here, conifers, cycads over here, dicots and monocots out there. Okay, I'm so sorry you're not able to see it. Okay, now ginkgos, knit-tails over here and conifers and cycads over here. So conifers, knit-tails, dicots, monocots, correct. Conifers, knit-tails, dicots, monocot. Knit-tails, dicots, monocots, conifers, wrong. So always remember monocot has to end. Okay, monocot is the last group of organisms that came up, okay, in the case of plants. So this should be the answer. From gymno to angiosperms. Okay, here dicots is given, this is wrong. Rest everything is same, just monocot dicot is being switched. Monocot is the one that comes up last. Now, replacement of the lighter color variety of peppered moth, that is Biston betullaria, to its darker variety, that is Biston carbonaria, in England is an example of, this is the previous year model question paper question. So, replacement of light colored moth, the white moth, to its darker variety in England is the example of natural selection, regeneration, genetic isolation, genetic drift. It is what? Natural selection. Okay, it is natural selection, nature is selecting it. At a particular locus, the frequency of allele A is, okay, this is new question. The frequency of allele A is 0.6 and the frequency of allele B, okay, capital A is this and small a is what? 0.4. What would be the frequency of heterozygous? That means you are asked about the frequency of capital A, small a. So ma'am, if A is this, if small a is this, we will just calculate 0.6 into 0.4. Is it? Does it give you the answer? No. Always remember, P square plus 2PQ plus Q square is equals to 1. You also write it as P plus Q is equals to 1. Actually, it should be P plus Q whole square, but this is also written somewhere, so do not be confused. Okay, anyways, I'll tell you when it is of use and when this is of use. Okay, let's go. P square, as in you are talking about the frequency of capital allele, that is denoted by capital P. That is denoted by what? P. That is denoted by the P letter. So P square means whenever you want to find organisms like this, you need P square. Whenever you need to find organisms like this, you need to do Q square. Okay, so over here, this is P and this is Q's value. This is P's value and this is Q's value. If you want P square, that means you are being asked about homozygous AA. In that case, you have to do A into A. If you are asked about homozygous small a, then small a, that is Q square. Here you are asked about heterozygotes, that means capital A, small a. So what do? You replace it. First comes 2, then comes P. P's value is 0.6, then comes Q. Q's value is 0.4. What will your answer be? Your answer will be 0.48. If at all you are asked in percentage, make it into percentage and write 48%. Here it's just not the percentage, the frequency is asked, so it will be 0.48. 0.48. Okay, now suppose, my dear children, if at all you are given the frequency of small allele, okay, is 0.4. What is the frequency of capital A? I do not know. In that case, use this. P plus Q is equals to 1. Where replace Q with what? Q with 0.4. And then is equals to 1. P will be 1 minus 0.4. P is equals to 0.6. P is equals to 0.6. And now they are asking you, not just this. Okay, they are asking you, what is the frequency of homozygous capital A, What do? You got the frequency of this. Do P square, that is 0.6 whole square. And your answer would be got. Okay, okay. That's how you need to use it. So please make sure whenever heterozygote is asked, do not forget to multiply it with 2. Okay, and homozygotes as in P square, Q square do. If at all in percentage, then convert into percentage. That's all. Chalo. Now, mention the evolutionary significance of the following organisms, shrews and lobfins. Lobfins will go first because we have studied about them. They mark a critical step in vertebrate evolution, bridging the gap between fishes and land-dwelling animals, as in what? Amphibians. These animals called lobfins evolved into the first amphibians that lived on both land and water. They were the ancestors of the modern-day frogs and salamanders. Frogs and salamanders are what? Amphibians. So they are ancestors of them, right? Yes. Now, shrews, my dear children, is the connecting link between reptiles as well as mammals and aves. It's a connecting link between reptiles. Okay, we'll just write aves. Reptiles and mammals. Remember, from reptiles you get mammals and apes, but over here, shrews is a connecting link between this and this. So shrews are among the earliest mammals and represent the primitive ancestors of modern mammals, providing evidence of mammalian evolution from reptilian ancestors. So basically, it is crucial in being the link between reptiles and mammals. That is the only thing that you need to write. So shrews here and lob fins there in between fishes and amphibians. Two things super important. Okay. Now, what are analogous organs? We already discussed it. Please go through it once more. Example of divergent evolution is also given. Okay, apart from what we studied, the thing that we studied is thorn of bougainvillea and tendril of cucurbita, right? Apart from that, you have limbs. The forelimbs of cheetah, us, bat. We all are mammals, my dear. Cheetah is a mammal, bat is a mammal, human being is a mammal. All of them have 4 limbs. But if at all you see, in cheetah it is used for running. In us it is used for eating. In bats it is used for flying. If at all you see the structures, the functions are very much different. If at all you see the structures, we all have the same structure. Humerus, radius, ulna, everything, carpals, metacarpals, phalanges, all of these structures are same. But we have divergent evolution. We have moved to different location. That is why we have moved to different locations, different habitats, which is why now the functions are different, but the origin is same. This is example of divergent evolution. The organs are homologous organs. Another example, you have vertebrate heart. We all have vertebrates, fishes, amphibians, all of us has heart, different chambers heart. One has 2 chamber, one has 3 chamber. Anyway, the structure is almost similar. We have atrium, we have ventricle, right? Again, that shows what? Divergent evolution. The common thing is the same only. It shows divergent evolution. Same thing with the brain also. Okay, so please remember these examples. Examples are super important. You will lose marks if at all you are not able to write it. Now, state Hardy-Weinberg principle. Explain any 4 factors affecting the Hardy-Weinberg principle. I already explained you some factors. We already learned about it, but let's see again. The principle says that allele frequency in a population are stable and is constant from generation to generation. What does it say? Allelic frequency. Okay, small t, capital allele, whatever allele is there, the frequency of alleles are going to be stable and constant from generation to generation. The factors affecting it, first thing, gene flow. Sum total of genes occurring in a sexually reproducing organism in a geographical area at a given time is known as gene pool. This is bigger explanation, you don't know how to write all this thing, okay? The transfer of genes between population of different gene pool which differs genetically but can interbreed is known as gene flow. The transfer of genes that is happening between 2 different populations out there. This results in the production of new combination. So isn't it affecting Hardy-Weinberg principle? Obviously yes. Genetic drift. It is a random changes in gene frequency in a small population. Purely as a chance is known as genetic drift. It is random, it is occurring by chance. Mutations, we know, a spontaneous and permanent changes in the genetic makeup that also brings out a lot of changes out there. Apart from that, you have natural selection. Natural selection also we learned. Selection of better adapted individuals with useful variations by the nature, that is known as natural selection. So if the nature is selecting this, the frequency of that allele will be more, right? Give examples and you can help explain them better. Now the last one which is there is genetic recombination. Over here, my dear children, when in sexual reproduction, what happens is that some part of our chromosomes get interchanged. Some part of our chromosomes get interchanged, my dear children, bringing about new changes, new variations out there, new combinations out there. That is known as genetic recombination. It is the process of formation during gametogenesis resulting in formation of new combinations of characters in progeny. So you can say during sexual reproduction or in sexual reproduction, we can see genetic recombination where the parts of the chromosomes are being interchanged, leading to what? Newer combination, changing the allelic frequency. So please remember there are 5 factors starting from here: gene flow, genetic drift, mutations, genetic recombination, natural selection. All of them super important. Done? Done. Now I have these 2 questions for you guys. Please go ahead, try to solve it from your end. Following diagram provides an example of, this is a diagram given, Australian marsupials in the middle and they have evolved. What is this an example of? And identify the correct arrangement of periods of Paleozoic era. So if at all I do not give you this question, I'm pretty sure you are not going to go ahead and go through Paleozoic era, Mesozoic era and all the eras. So go through it, super important. Okay? Yes, and let me know. So I think that's all. Thank you so much for being a part of Brahmastra series. I hope this series is helping you well. If at all it is, my dear children, do not forget to share it with your friends also, because you have the exam season going on, they need help, right? And if it helped you, do not forget to hit the subscribe button. And that's all. Take care of yourself. Bye-bye.